Hi everyone, I'm Julie Daniels, Professor of Regenerative Medicine and Cellular Therapy at the UCL Institute of Ophthalmology, where I work very closely with colleagues at Moorfields Eye Hospital. So welcome to this talk on the future perspectives of limbal stem cells. Of course, I'm sure many of you know that the cornea, our window to the world, is comprised of three layers that contain cells. On the inside layer is the endothelium, which helps maintain corneal transparency by maintaining an appropriate water content. The majority of the cornea is made up of a transparent stroma, which is largely composed of collagen. And the outermost layer is the epithelium, which is a little bit like the epidermis of skin. And this is the one that we're going to be focusing on today. So, of course, every time we blink, cells are lost from the surface of the cornea, and these cells need to be replaced for us to maintain a healthy epithelial tissue. And there's a population of adult stem cells known as limbal epithelial stem cells, which are responsible for producing daughter cells that maintain our corneal epithelium throughout life. So if you imagine a ring drawn around the outside of the cornea, this is a location called the limbus, and this is where we can find these limbal epithelial stem cells. And they predominantly reside in the 12 o'clock and 6 o'clock positions. So typically they're covered by the eyelids, and we think that's not by chance, and it's probably there. They probably find themselves there because it's a nice niche or home for the cells, which is a protective environment from, for example, from UV damage. So when we think about aniridia, some patients develop a condition known as aniridia-related keratopathy. And as we know, aniridia is related to various mutations in the PAX6 gene. And one of the consequences can be a failure of these limbal stem cells to work properly. So it could be known as limbal stem cell failure, limbal stem cell deficiency. And essentially what happens is the cornea can become cloudy, neighboring conjunctival tissue grows over the surface, bringing with it blood vessels. So we've got the wrong tissue in the wrong location. And there have been a number of surgical approaches to attempt to reconstruct that corneal surface to make it more normal-like again. But one of the quite exciting developments in the late 90s, which was the work of Graziella Pellegrini and colleagues in Italy, was to actually take a population of these limbal stem cells into the laboratory and culture them. So we had many more of them. And with them and their daughter cells, they can form sheets of epithelium which can be transplanted. And we and other colleagues have used this technology in an attempt to address the ocular surface problems associated with aniridia-related keratopathy. And for some patients, they were able to have an improvement in their ocular surface, which manifested as improved vision for up to 3 years. However, other patients didn't get any benefit or very little benefit, or that benefit was short-lived. So it got us thinking about why might that be? And our hypothesis was that we were growing normal, healthy limbal stem cells in the laboratory from donor tissue. So we were fairly confident that those cells were of good quality before they were transplanted. But it may be the equivalent of planting those healthy stem cell seedlings into poor quality soil, as in the recipient stroma of the patient with PAX6 mutation may not be optimal to receive and to let those cells thrive. So our idea is to combine those epithelial cells with stromal cells, so cells that normally live in that in the collagen of the cornea, and to provide that in a natural matrix in the hope that this may be a more superior therapeutic option. And the matrix that we've chosen is collagen because that's the main protein of the cornea. So in order to be able to develop this potential new therapy, we needed to understand a little bit more about the home where these stem cells normally live. And this home is referred to as the stem cell niche. And when we specifically look in the 12 o'clock and 6 o'clock positions that I was referring to earlier, we can actually find structures that we've termed limbal crypts. And these crypts are full of epithelial cells. And at the base of these epithelial cells, there are very, very small cells, and they have features. They look like cells that we would typically associate with stem cells. So they're right at the bottom of these crypts. And interestingly, in very close proximity, we noticed another cell population, which we're calling stromal cells. And we wondered all those years ago when we first observed this, whether these stromal cells may play an important role in maintaining the limbal epithelial stem cells. And in fact, further work suggested this might be the case. So when we look in these crypt-rich regions at 12 and 6 o'clock compared to non-crypt-rich regions at 3 and 9 o'clock around the cornea, we see interactions between the cells that we think might be stem cells with a stromal cell, and also another cell type, a melanocyte, which produces pigment to help protect those epithelial cells from UV damage. So the stem cell niche is actually quite complicated. And what's really good news is that if we target these niche areas that we know are rich in these putative stem cells, and I keep saying putative as in they are likely to be stem cells, but we can't say for sure because there are still yet no really specific markers for them. So our evidence is quite indirect. But if we take cells from these niche regions into the laboratory and expand them in culture, we can get really large colonies, which are known as holoclones. And these are thought to be the best representation in the laboratory, at least, of a stem cell. So we were able to culture them, which is really good news. So with this information and lots of other research as well besides, we wondered if perhaps we could develop a new therapeutic option. And to do this, we worked with Professor Robert Brown, a tissue engineer at University College London, and he and his group had developed a tissue engineering method known as plastic compression of type 1 collagen. And essentially what this is, you can take type 1 collagen that's been extracted and make a jelly-like material out of it. And when you're making it, you can put cells of your own interest in there. So in our case, the corneal stromal cells go inside, and then this gel gets compressed between absorbent material, and over time, around about 15 minutes or so, water is extracted and you end up with a tissue-like material. And when we actually look at these materials under the microscope and we stain them for markers, what we find is that when we put the corneal stromal cells inside the collagen gel, we get a much better quality epithelium on the surface than if we leave those cells out. So without the stromal cells, we get quite a disorganized epithelium, we get holes, and they just don't grow as well. So this backed up our observations of what was going on in the normal stromal stem cell, in the normal stem cell niche, and we thought maybe putting these two cell types together is going to be beneficial therapeutically. And when we look at the quality of the epithelium, we find many small cells that express a possible stem cell marker called p63 alpha, and also the daughter cells that need to mature, the ones that are constantly sloughing from the surface of our eye, express a protein called cytokeratin 3, which is associated with a corneal phenotype. So far, so good. But the way that we were making these tissues in the laboratory was very fiddly. It wasn't very reproducible because they were all being made one at a time by hand. So we started to work with a company called TAP Biosystems. And the idea was that we were going to together develop a machine that would be able to make these tissues for us for use in the cornea. And actually, we got down to a much simpler solution, which was a set of disposable consumables where we could make these tissues instead. So the process is now known as real architecture for 3D tissues, or RAFT for short. And as a side project, this technology then went on to be taken over by Lonza. So there are loads of people using this now for assay development, drug screening, tissue models, cancer models, etc., because it's a very highly tunable way of making artificial tissue, and it's also much more reproducible than when we were doing it before by hand. So coming back to the cornea, so what are we doing, or what have we been doing to move towards a new corneal therapy? Well, the first thing that we've had to do is to actually make a bank of these stromal cells. So we would take tissue, or rather we took tissue from an eye bank, and we have been able to extract a population of corneal cells, which have also themselves been proposed to be stem cells actually, and they divide many times. We can get lots and lots of them. And we learned how to do this from our colleague Jim Funderburg at the University of Pittsburgh. So we now have frozen down many vials of these cells ready in advance to be used in a therapy. But the stage that we froze those cells down at, they weren't ready yet for therapeutic application. So the next thing that we do is, when we thaw some of these cells, we then put them into a different culture media, and we provide a different environment, which makes these cells change to become more like the cells that you would normally find in the cornea. And they're called keratocytes, and they live between the collagen layers of the cornea. And we had to be really sure that these cells were not going to cause any problems for patients, because following injury, these cells can become activated and become involved in a scarring response. So we actually used a model where we transplanted our collagen raft with these keratocytes inside, and then made observations over a period of a month. And we were able to find that we didn't see any inflammation, we didn't see any blood vessels growing across, and we didn't see any scarring, which was all good news for progressing this forwards. So from there, we then had to look at the whole manufacturing process. So this new therapy we refer to as raft ocular surface, which is our collagen with the keratocytes inside. Now we need to get the epithelial cells on the surface. So again, we take a piece of tissue from a donor, from an eye bank, and we chop up that tissue into small pieces. And then when we stick that tissue onto the top of the raft, the epithelial cells grow out. And as they grow, a wave, an edge can be seen. So over time, we can see for ourselves when the whole surface is covered and when it's ready to go to a patient. Then we have to package it all up, which sounds maybe quite trivial, but we have to go through lots of validations to make sure that we can get this very carefully prepared material to the surgeon in the appropriate way, and so that it's, it doesn't lose any of its quality. So it's still going to be a useful product when it gets there. And there are very specific labeling requirements for the regulators. So with all of this in mind, so we've got our preclinical data, our validation data, which is all about manufacturing. We had to generate an investigator's brochure, which talks about all our historical research leading up to this point, an investigational medicinal products dossier, which explains how we're going to make the therapy, a clinical trial application describing what will happen to the patients, and of course, ethics approval. So I'm very pleased to say that all the regulatory approvals are in place, and we are now ready to begin our phase 1/2 clinical trial. This is funded by the Medical Research Council's Developmental Pathway Funding Scheme, and it's being led by my colleague, Dr. Sajjad Ahmad at Moorfields Eye Hospital. The primary outcomes for the study are primarily, number one, safety, of course, And number 2, efficacy. And by that we mean, is there any normalization of the corneal surface at 3 months? And our secondary outcomes will be any improvement in visual acuity at 3 months and 12 months. And of course, very importantly, asking the patients for their assessment of whether their quality of life has been improved, or not. So we hope to recruit 21 patients through Moorfields Eye Hospital from the summer of 2021. I'd just like to acknowledge all of the various funding agencies that have supported our work over the years, and of course, all of our friends, colleagues, and team members that have contributed to this project. And I think that it's not an exaggeration to say that around 100+ people have worked on this RAF project to bring it where it is. So thank you very much for your attention, and I'm happy to take questions.